everyone. Thank you so much for joining us for the LabRoots 2025 Neuroscience Virtual Event Series. My name is Jie Zheng. I am assistant professor from Department of Neurological Surgery and Department of Biomedical Engineering at UC Davis. I started my lab last year at UC Davis. My lab, or Z Lab, we interested in understanding the neuron mechanisms underlying various of cognitive functions in humans, including learning and memories. And we use very comprehensive tools to understanding that brain process, including in-vivo electrophysiology recordings in humans and eye trackings, behavior measurements, and also computational models. Today, I'm very honored to be here to share some of our recent findings about event segmentations and how that be served as potential cognitive markers for patients with neurodegenerative disease. So before I started introducing the experiment and also the results we were doing, I want to introduce the process of event segmentation first. The event segmentation is a very fundamental memory process that happens every day. As we all know, we experience the world continuously. However, our memory is not continuous. So we cannot remember every single thing we have experienced, but instead, we have those discrete memorable moments or events we can extract it from our continuous interaction with the world. For example, for a road trip we have went, we won't be recall every details during the road trip. However, our brain was able to capture those memorable moments. For example, the fun night we spent in the camp, or those story nights we have seen during the nights, or those beautiful things we have observed with our friends together. So clearly, our brain is constantly taking this kind of mental snapshots from our continuous experience and store them in the memory books we have and reorganize them for future use. And this process of event segmentation has previously been shown rely on the event boundary detection. So event boundary is a very critical indicators for the process of event segmentations. It helps us to identify the transition of different events with different contextual contexts, or help us to organize our memorable moments or the events in a structured way that help us to facilitate the later memory recall. The impairment in the ability of detecting those event boundaries or the impairment of abilities of event segmentations can lead to a lot of memory problems. For example, if we detect too many event boundaries or we are over-segmenting our events, that will lead to very fragmented memories, making our ability very hard to recall the continuous or cohesive memory representations of our experience. In opposite, there's cases maybe the brain was detecting too little or too few event boundaries, or in other words, we are under-segmenting our events from our continuous interaction with the world. In this case, will lead to very disorganized memories or oftentimes memory interference and also impair our memory abilities. And such segmentation or under segmentation or the impairment of boundary detections often appeared in the patients with neurological disorders and also in aging groups. And such impaired event segmentation abilities can also lead to impaired memory performance in those groups. In the study published by Gabriel Kreiman and his colleagues in 2026, they specifically studied such differences in terms of event segmentation behaviors in the elderly groups who are diagnosed with Alzheimer's disease compared to the young generations. In this task, they presented a series of clips which consist of a sequence of day-to-day events, and asking the subject to make an annotation whenever they think the event boundary occurs. They then compare the event segmentation behaviors between these two groups. As shown in the results here, the lighter bar indicates the results from the young groups, and the darker gray bars are showing the results from the older groups. The first thing we're looking at is the mean unit length, which means how much the event segmentation they did and how long each segmented events they are. As shown on the plot here, we can see that the older groups tend to have a longer event segmented from the same clips they have watched compared to the young groups. That means they're probably experiencing some effects, some impairment in terms of under segmentation, as we mentioned earlier. And after they segmented all the events, subjects are also asked to perform a memory tasks, which they need to recall the specific order of those segmented events from the clips they have watched. As showing on the right plots, older groups have difficulties recalling the older memories from the events they have been presented in the clips, with larger order memory errors compared to the young groups. And in this study, no matter whether the experimenter explicitly telling the subjects whether there's a memory task in the following they watch the clips or not, such age group differences was also observed between the young and older groups, with consistently findings showing that the older groups they tend to under-segment the continuous experience, also lead to a very worse order memory performance compared to the young groups. So clearly, it shows that event segmentation ability or the ability of detecting event boundary is impaired in the aging groups or groups with neurodegenerative disease. So the next question is, what is driving these event segmentations and how the brain is facilitating this process? As a popular theory proposed by Dr. Jeffrey Sachs, or so-called event segmentations theories. Dr. Sachs proposed that our brain is constantly making prediction using the current information or sensory inputs received and also our event models stored in our brain. And when our predictions is violating what we have actually received during the actual experience, there will be prediction error generated, which can also be detected by the brain and used as a feedback signal sent back to updating our current event model. So for example, if you're attending a birthday party, if you see the cakes coming out and also with the candles lit, you can automatically predict it's about the time to sing the birthday song. However, if suddenly a dog comes in and trying to take the birthday cake away, this is violating what you're expected. And your brain will be able to capture that prediction error and updating the event model right now, trying to rescue this party and comes up more solutions to save the birthday party. So it seems like the prediction error detection is the key driver here that helps the brain and also associated with event segmentation or event boundaries here. Then what neural signal is actually associated with this prediction error detection? One beautiful work did by Dr. Schaus in 1997 from the science paper they published. They built these connections between this prediction error detection with the neural activity from the dopaminergic neurons they recorded. So in this experiment, they trained the monkey to build association between the cue and also a reward. What they found is when the monkey can predict the reward, which means the reward was presented followed by the associated cue, dopamine neurons do not respond to that very differently. However, In the situation when the reward is unpredictable, for example, in the case the reward was presented without the sound or associated cue was presented, or in a different case, when the associated cue was presented, somehow the reward is not given to the animal. The dopamine neurons recorded in the monkeys either increase their firing rate or decrease their firing rate in response to this unpredicted reward. So building upon these findings, between the links between the prediction error detection and also dopaminergic neuron responses in the monkeys, we wonder whether dopamine neurons also responded to the event boundaries as a different type of prediction error as well. To address this question, we developed a memory tasks, consistent with two different phases. During encoding, we were presenting a series of clips to the subjects. So the clip itself doesn't have any audio inputs, and it's just a series of videos. And at the end of each clip presentation, we ask the subject to answer the question whether the majority of the clips happens indoor or outdoor to keep them focused during this passive viewing period during the encoding. And the key manipulation here is we have two different types of clips. The first one, we name it as clips with virtual boundaries. Here is one example you can see. So in these clips, you can clearly see the man was making the dough, preparing the dough first and putting into the oil for further cook. So there are two events. But the event transition wasn't marked by any manual editings. Instead, the clips with virtual boundaries is actually a continuous shot from the same movie. We also have a different type, which name it as a clips with event boundaries. Here's one example of that. So in this event, you can see that it was transitioning from the girls practicing boxing and to the men's waiting in the bus station. And these two events are pulling out from two very different contexts and doesn't have a very clear contextual relevance. And we refer these event transitions in this type of clips as event boundaries. So during the entire encodings, subjects are watched 60 of these clips with 30 event boundary clips and 30 virtual boundary clips. After a short break, and the subject will be instructed to perform memory tasks in which we extracted the frame from the clips they have watched during the encoding period and asked them whether it's old or new, or sometimes we extracting the frames from the clips they haven't been shown during the encoding. So that will be the novel images. And we are running this task in the patients undergoing deep brain stimulator implantation. So it was performed intraorally. And these patients usually are undergoing this kind of surgery to implant a deep brain stimulator to help with their motor disorder symptoms. And they will be wake up during the surgery to help mapping the motor functions in order to find the best location for the implantation. And we are leveraging these precious opportunities during the OR and trying to run our behavior tasks here by simultaneously collecting subjects' behavior responses about the memory tasks and also recording their neural activities. To record their neural activities, we are using the typical standard clinical probes, which the clinicians are used to targeting the brain regions for deep brain stimulator implantations. And this device, on the tip of the electrodes, it has the microchannels, which is capable recording deep neuron activities extracellularly. As showing on the right side in the bottom, that will be the typical single neuron actual potential we have recorded from the extracellular recordings using this type of probe. And for this study specifically, we are very interested in the neural responses from substantial nigra, which is the brain region from the midbrain that was known for producing and releasing dopamine signals. And this is also a routine entering spot for the subthalamic nucleus implants, which we usually get from the DBS recordings intraoptically as well. So the first thing we were trying to understand or testing is, since the test was done in the operating room and the patient is waking up just from anesthesia, we want to make sure they can handle the task with reasonable behavior performance. So we run the task not only on the patients undergoing DBS implants, but also we run it on the healthy control groups outside of the operation rooms. And we want to compare their behaviors in terms of the questions we asked during encoding, whether the majority of the clips happens indoor or outdoor, or their responses during the retrieval to test their recognition memory task. So in both groups, we can see that they have the above chance level encoding accuracy and also the recognition accuracy. Even though the patients under DBS implants, they have slightly lower recognition accuracy compared to the healthy control group, but it's still way above the chance level, which is 0.5 here. Means that the patients undergoing DBS implants can actually handle this task. And within the patients undergoing DBS implants, if we take a closer look at their memory behaviors during the recognition period by separating the trials, whether the images are coming from event boundary clips or virtual boundary clips, it seems like images from event boundary clips are better remembered compared to the virtual boundary clips. Further verifying our hypothesis that the event boundary actually can help people memory in general. So at the neural level, we also want to see whether there's neurons actually responding to those different boundaries we embedded in the clips. As shown here, we align the neural activity to 0, so either it's the onset of event boundary or virtual boundary color-coded differently for those two different conditions. And this is one example neuron we have found in the substantial nigra, with each dot representing the activation of that specific neurons and color-coded differently for these two different conditions. As shown here, this neuron increased firing rates following the event boundaries against the baseline, also against the virtual boundaries. We also find another type which they suppress their firing rates following the event boundaries against the baseline, also against the virtual boundaries. And among all the subjects we have recorded, we have observed 22 this and also verse 14 this kind of boundary cells in the substantial nigra either increase their firing rate or decrease their firing rate in response to the event boundaries during clip watching. And the next question is how the neuron behave when they are retrieve their memories, when they are presented with the novel or familiar images during the retrieval tasks. So now we align the neural activity to the novel image or familiar image onset. As shown here, this is one neuron we recorded from the substantial nigra with increased firing rate following the novel image, compared to the baseline, also compared to the familiar images. And similar to the boundary neurons we have observed, there's also types of substantial nigra neurons decrease their firing rates compared to the baseline and the familiar images when they're responding to the novel images in the retrieval tasks. And across all the subjects we recorded, we have observed 27 memory cells and 18 memory cells that either increase or decrease their firing rates against when responding to the novel image against the baseline versus the familiar images as well. And interestingly, among those boundary cells and memory cells, we found a huge overlaps between these two groups. And if we look at the overlap neurons between these two groups, and plotting their firing rates at boundaries during encoding or at novel images during retrieval, their firing patterns are very similar, with their firing rates highly correlated for both boundary and also novel image presentation. So between this corresponding to the boundaries and also the novel images during retrieval, we also wonder whether those overlapped neurons contributing to memory performance in humans as well. What we did is we extracted the trials, we separated the trials based on subject performance during the memory retrieval. Either they correctly recognized the frames or they didn't. And then we computing the firing rate at boundaries within those two different type of trials, and also their firing rate at the image display. What we see here is those overlap neurons tend to fire a little bit more when the subject correctly recall recognize the images during the memory retrieval, when they are the firing rate at the boundaries and also at the image display, compared to the trials when the subject forget or didn't recognize the image. And such memory effects or differences in terms of firing rates of those overlap neurons for both boundaries, at boundaries and at image display doesn't exist or doesn't generalize to the virtual boundaries or the familiar images. So how come those overlap neuron or the neuron responding to both boundaries and novel images are also contributing to subjects' memory performance? I think our studies actually provide the neural evidence to the Lin and Grace models for long-term memory. So in the papers Lin and Grace published in 2025, they propose there's a functional loops between hippocampus and midbrain dopamine system, including substantial nigra. In this case, hippocampus was constantly predicting what's going to happen next by integrating or controlling the sensory inputs. And once the hippocampal prediction violates what actual situation it is, the prediction error will be sent to the substantial nigra, our midbrain dopaminergic regions. And in return, substantial nigra will release dopamine and going back to the hippocampal regions to enhance the neuroplasticity and also enhance the encoding of the novel situation or unpredicted situations into our long-term memory system. So we clearly also see some of this evidence in our hippocampal recording as well. In different tasks, when we are running a similar experiment on patients with drug-resistant epilepsy while recording their activities at the single cell levels from the hippocampus, we also find some of the neurons in the hippocampus responding to this kind of boundaries signal as well. And interestingly, those boundary neurons we found in the hippocampus, their firing rates when responding to the boundaries is also predictive of subjects' subsequent memory performance. So during the scene recognition memories, if the neurons fires more at the boundary during encoding, that associated with a correct recognition behavior later. In summary, what we show here is the event boundary detection is a very critical abilities people are using day to day. And oftentimes this ability is impaired in patients with neurodegenerative disease. And we observed that neurons in the substantial nigra actually responded to this kind of event boundaries with either increased firing rate or decreased firing rates. And also, we observed those most of boundary neurons observed in the substantial nigra was responding to the novel images. We believe both boundaries, event boundaries, and novel images are reflecting the prediction error signals and predicted or consistent with the prediction from the Lin and Grace models. The boundary neurons firing rates following event boundaries and novel images predicts participants' recognition memory. And building around all the findings from the behavior studies, the neuroimagings, and also the intracranial recordings, such as the results from our group, we hypothesized that this might be the moment for integrating all the findings from the basic neuroscience study to moving forward to translational applications by building a closed-loop neuromodulation system. For example, we can receive the scenes people have seen through the first-person cameras and using computational models to make a decision of event boundaries. When the suboptimal event boundary responses was detected from the brain signals, then we can apply the electrical microstimulations to help the neurons or help the brain identify the correct event boundaries and also enhance their memory structure and enhance the memory behavior in the long run. And this study is a team science and a product. And I want to thank our collaborators from Beijing Tiantan Hospital, especially Dr. Ling Shi, who helped us collect the data and with a lot of the fruitful discussions in terms of these projects. And also I want to thank the graduate student and undergraduate student research assistants who helped analyze the data for this project. And also our amazing clinical teams supporting such research projects in the operating rooms, and our lovely patients, and our funding agency, NIH Brain Initiatives. So I want to thank you as well for your attention. And if you have any questions or interested in other studies done in the lab, please feel free to reach out and also visit our website, zlabcognition.org. Thank you.